The overall goal of this procedure is to reconstitute mitotic spindle-like structures within the geometrical confinement of water in oil emulsion droplets.
This method can help answer key questions in the mitotic spindle assembly field, such as how are spindles positioned and assembled, and what is the specific contribution of individual components to these processes.
The main advantage of this technique is that we use a bottom-up approach to reconstitute spindle-like structures within the geometrical confinement that mimics the shape of a mitotic cell.
This method can also be used to study other processes that are dependent on cellular confinement.
Mix 10 parts PDMS pre-polymer with 1 part of curing agent in a total mass of about 40 grams. Then, place the mix in a vacuum chamber for about 30 minutes to remove air bubbles. Meanwhile, wrap aluminum foil around the mold to create a 1 cm deep cup with a 4-inch diameter. When ready, pour about 3/4 of the PDMS mix into the mold. Then, return the PDMS to a vacuum chamber for another 30 minutes. Following degassing, cure the PDMS for an hour at 100 degrees Celsius. Meanwhile, prepare some glass slides for spin coating by dusting them off with compressed air. Then, spin coat the remaining PDMS mix onto the slides. Cure these slides for an hour at 100 degrees Celsius to harden the PDMS.